Hi, I'm Sam, a pharmacologist and a medical science liaison. Welcome to the very first episodes of How Medicines Are Born. We all have experienced using medicines in our lives, but have you ever wondered how these medicines came to be? Today we are starting an exciting journey from the initial spark of a scientist idea to the life-saving medicine on your pharmacy shelf. In this episode, we will explore the detailed process of drug development, including challenges, success, and real-life stories behind these important treatments. Today, we are embarking on a journey from the ground up, from identifying an unmet medical need to discovering a molecule that brings hope. And along this journey, we will dive into one of the most inspiring stories in modern medicine. Discovery of imatinib for treatment of chronic myeloid leukemia, CML. Are you ready? Let's begin. Every story begins with a gap. Our story starts with identifying the unmet medical need. Imagine this: a quiet morning in a busy research center. In a conference room, charts show the grim statistic of chronic myeloid leukemia. Before 2001, where imatinib was approved, the life expectancy of these patients was very short. The only definitive treatment available was bone marrow transplant, a risky and expensive option, which was not available for most. In that quiet room, a simple question was repeated: can we find a better option? Can we find a better treatment? That simple yet powerful question sparked the research for an unmet medical need. A call for new ideas where the hope was low. The second step is identifying the therapeutic target, uncovering the root cause. Once the need was clear, scientists set out to understand the disease at its core. We can explain this further with our imatinib example. In 1960s, researchers discovered an unusual change in cancer cells of CML patients. A translocation between chromosome 9 and 22, famously known as Philadelphia chromosome. This abnormality produces a harmful protein called BCR-ABL, a tyrosine kinase that is permanently switched on and continuously pushing cells to grow and divide. The challenge was clear: develop a drug that could selectively block this overactive protein without disturbing body's normal function. This clear target laid the groundwork for one of the most important advances in medicine. The third step is screening. Screening is like finding a needle in a big pile. After finding the therapeutic target, researchers jumped into one of the most complex phases, which is screening. Screening millions of chemical compounds using high throughput or HTS technology. Every molecule was tested carefully to see if it could bind to the unwanted protein, inhibit its harmful activity, and do so selectively, leaving body's essential pathways unaffected. Out of millions, 99.9% of the compounds were eliminated for being ineffective, toxic, or unstable. But their hard work paid off. In that vast sea of data, one molecule emerged: a selective tyrosine kinase inhibitor that exactly targeted BCR-ABL. This discovery paved the way for imatinib, a drug that soon would change lives. The next step is preclinical evaluation. After screening, the selected molecule was tested on cells and animals in preclinical studies. These studies answered important questions. Does it work as intended? How toxic is it? How is it absorbed, distributed, and eliminated by the body? What are its overall biological effects? Imatinib shined in this test, demonstrating high efficacy, low toxicity, and excellent stability. This success cleared the path for the next bold step, human trials. The next step is the first human trial. The first human trial is a nerve-wracking moment in medicine. While phase 1 trials often start with healthy volunteers, in life-threatening conditions like CML, patients become the first to try new treatments. In these trials, safety, tolerability, and finding the optimal dosage are top priorities. Every heartbeat and every moment is carefully watched. I can imagine how tense the room must have been when the first patient received imatinib. In that quiet moment, all eyes were on the monitors. And yes, the levels of the cancer cells began to drop and the treatment started to show promise. This was a moment when hope moved from the lab into real lives. Today we have followed the path from a need for better treatments to finding a clear target, to testing many compounds, and then doing detailed lab and patient studies that led to imatinib. But this is only the beginning of our journey. In future episodes, we will take a closer look at clinical studies from phase 1 to phase 3 and share real human stories. If you are curious about the stories behind every medicine, join me on this journey. I'm Sam, and thank you for listening to How Medicines Are Born.